Hi guys, so today we're going to talk about biomolecules, and these are very important not only for this class but for your life in general, because these are the molecules that allow us to exist. So this lecture is going to be really good practice for you guys to start breaking down words into their roots, prefixes, and suffixes, because if you can do that, that usually will tell you just what the word is talking about. So let's take a look. Biomolecules. Bio means life, like biology is the study of life. So these are the molecules of life. Now they are non-living molecules in themselves, but they allow bigger organisms like us to live. And there are four major classes of biomolecules: carbohydrates, lipids, proteins, and nucleic acids. So we're going to get into each of those in a bit, but before we do that, there are a few concepts that you need to understand first. The first of which is dehydration synthesis. So let's think about what the word dehydration means. When you're dehydrated, that means you have lost water. So dehydration synthesis is the process of losing water to bring two smaller molecules together. See, out pops our water molecule here. Make a bigger molecule. Now the opposite of dehydration synthesis is a process called hydrolysis, where a water molecule is inserted into a larger molecule to break it down into two smaller molecules. Now all four classes of biomolecules use those two processes, and we'll see examples of that as we go along. So here are some really common prefixes that you should know. Mono means one, di means two, tri means three, and I forgot to include it, but poly means many. It would also behoove you to know some of these functional groups over here. This will also help you with your chemistry classes. So feel free to pause the video and study this as you so desire. It is also good to know the bonding patterns of some of our most common elements in living things. So carbon likes to form four bonds. See, 1, 2, 3, 4 surrounding this carbon here. Hydrogen likes to form one bond. Notice one bond there. Oxygen likes to form two bonds. Notice there's a double bond there. Or even down here, we look, there's 1, 2 for oxygen. And nitrogen likes to form three bonds. 1, 2, and 3. It is also assumed that you know that in organic chemistry, organic chemists don't always draw the carbons on their molecules. So wherever a line meets another line, it's assumed that you know that there is a carbon there. So this molecule is the same as this one over here, even though there aren't carbons drawn as they are over here. Okay, so let's start talking about our first class of biomolecules, and that is carbohydrates. So let's break down this word. Carb, carbon, O, oxygen, hydrate, hydrogen. So there are three types of carbohydrates. We have our fiber. Now fiber is something that humans cannot really digest in the diet because we don't have the enzymes necessary to do so. However, it is still very important in our diet because the fiber helps bulk up our stools, which keeps our intestines healthy, and it also helps lower bad cholesterol. There are two subtypes of fiber, soluble and insoluble. Next we have our starches, and starches are big long chains, or in other words, polysaccharides of some of the smaller sugars that we are going to talk about in a bit. So there are two different kinds of starches, amylose and amylopectin, but we don't talk all that much about them in this class, so let's jump down to the sugars. So whenever you see the root word saccharide, be thinking sugar or carbohydrate. Also, all of our sugars have this OSE ending on them. For example, Down here we see glucose, fructose, galactose, deoxyribose, and ribose. So start to look for those kinds of patterns. Now all of our biomolecules can be kind of broken down into smaller subunits, and those are something that we call monomers. So for sugars, the monomers of sugars are mono, one, saccharide, sugar. Makes sense, right? So let's look at our monosaccharides. The big three that you need to know are glucose, fructose, and galactose. We also see deoxyribose and ribose, but we're going to talk about those with the nucleic acids section, so put those on the back burner for a little bit. So, glucose, fructose, and galactose all have the chemical formula of C6H12O6. That means 6 carbons, 12 hydrogens, 6 oxygens. If you were to count that out in each of these molecules, you would indeed find those number of elements. However, you do notice that these are different molecules, so that brings us to the concept of isomers. Isomers are molecules that have the same chemical formula, but the elements, or atoms, are arranged differently in space. So let's take a look here. Here's our glucose. We know it has 6 carbons, so let's count them out. I see 1 here, 2, 3, 4, 5, and 6. Now let's count them out in fructose. I see 1 here, 2, 3, 4, 5, and 6. However, glucose is said to be a 5-carbon ringed structure, but with 6 carbons total. And then we have this one on the branch chain here. Fructose is said to be a 4-carbon ringed structure with 2 side chains containing carbon. And galactose and glucose are very similar, but if you notice over here on these side pieces that the hydroxyl groups and hydrogens have been flipped. So glucose is our especially important monosaccharide because glucose is what our cells use to turn into ATP, which is the energy currency of the cell. So when we eat other types of sugars, or any carbohydrate for that matter, your body will turn them into glucose and then start the process of cellular respiration, which is a whole different conversation. Let's look at our monosaccharides in your lab manual now. So you need to be able to recognize all these different biomolecules here in these structural formulas. So let's try to find, what do we have here? Fructose and glucose. So I see a ring, so immediately I think this might be one of them. So let's go ahead and count it out. See a carbon here. So 1, 2, 3, 4, 5, 6 carbons total, 5 of which were in the ring, so that tells me that this must be our glucose. I see another ring shape over here, so let's count these out. See 1 carbon, 2, 3, 4, 5, and 6, but there are only 4 carbons in the ring with 2 side chains for a total of 6, so that tells me this must be our fructose. So now you need to be able to kind of transpose these images onto the 3D molecules that you see in lab. So pay attention to our color key over here. Notice that our carbons are in black. So these can be a little bit tricky, but just count carefully and you can do it. So I see a ring structure over here. So let's count out the carbons, the black balls. So I see 1, 2, 3, 4, 5, and 6. Notice the ring shape here. So 5 of those carbons were in the ring, which tells me that this must be glucose. And if you were to count out all these oxygen and hydrogens as well, you would find that there are 12 hydrogens and 6 oxygens. See another ring shape over here, so let's count this out. I see 1, 2, 3, 4, 5, 6 carbons total, 4 of which are in this ring structure here, 5 and 6. So that tells me that this must be fructose. Alright, so now let's look at our disaccharides. So remember, our prefix di means 2. So these are just 2 of the monosaccharides we just talked about that are bound together via dehydration synthesis. So here's a reminder of what that is. We take a hydroxyl group from one monosaccharide and a hydrogen from the other. 2 H's and 1 O, H2O. Out pops a water, and we went from 2 monosaccharides to a disaccharide, with the oxygen sort of forming a bridge in between the 2. So the 3 disaccharides that you guys need to know are sucrose, which is table sugar, which is composed of a glucose and a fructose. Then we have lactose, which is a milk sugar, which is composed of galactose and glucose. And then we have maltose, which is composed of 2 glucoses. Notice that we still have that OSE ending on all of these names. So let's look in your lab manual and see what we can find. So here I see 2 rings. So let's kind of count out what it might be. And our options over here are maltose and sucrose. So you see 1, 2, 3, 4, 5, 6 carbons total. So that's good. And 5 of these were in the ring. So that tells me that this is a glucose. And over here we have 1, 2, 3, 4, 5, and 6, 5 of which were in the ring, so that tells me this is another glucose. So glucose plus glucose makes up a maltose molecule. I don't see any other double rings on that page, but here's one over here. So let's count this out. See 1 carbon, 2, 3, 4, 5, and 6, 5 of which were in the ring, so that tells me this is a glucose. Then over here I see 1 carbon, 2, 3, 4, 5, and 6, but only 4 of them were in this ring, 2 were on the side chains. So that tells me that this is fructose. And remember that a glucose and a fructose together make up sucrose, which is the white table sugar. Now let's take a look at polysaccharides. Poly means many, saccharides, sugar. So these are just big long chains of those monosaccharides that we were just talking about. Starches and fiber that we talked about earlier fall under this category. But the big one to pay attention to for this class is glycogen. So glycogen is a big, long, branched chain of glucoses. And glycogen is a storage molecule in our bodies. So when you eat a food that has a lot of sugar in it, your blood sugar levels rise, and your body kind of wants to save some of that glucose for a rainy day or a time when you might have less energy, like when you're exercising or when maybe you haven't eaten for a while. So it will take some of the glucoses out of your bloodstream and start to build this glycogen molecule out of it. And the liver is the main storage space for glycogen. Skeletal muscle also stores glycogen, but the glycogen it stores is used only for the skeletal muscles, whereas the glycogen that is stored in the liver can be used as energy for any cells in your body. So, when you are low on blood sugar, your body can sense that, and it tells your liver to start chopping off little glucoses from this big glycogen molecule and sending the glucose out into the bloodstream so that the rest of your cells have energy. So let's find our glycogen molecule here. And this is it. So each of these little, they kind of look like circles here, are supposed to be a glucose. Now we can talk about some of the hormones that help control blood sugar levels. So both of these hormones, glucagon and insulin, are made in the pancreas. And glucagon is released when blood sugar is low. So glucagon stimulates the liver to start breaking down glycogen to release glucose so that your blood sugar level raises. On the flip side, insulin is released when blood sugar levels are high, and that tells your cells to, hey, uptake this glucose to make some energy with it, and it also tells your liver, take some of this glucose out of the blood, save it for a rainy day, and turn it into glycogen. Alright, so now we can talk about our next class of biomolecules, the lipids. So lipids are nonpolar molecules, which means that they will not interact with water, or they are not water soluble. Now, water is a polar molecule, and water solubility is kind of a whole other conversation, although I do highly encourage you to look into that. So, since lipids are nonpolar, that means we can call them hydrophobic, or water repelling, or we could also say they are lipophilic, which means they're attracted to other lipids. So lipids make up our fats and our oils and our butters, kind of on the macroscopic scale, and inside the body on a microscopic scale, lipids have a variety of functions, which we will see as we go along here. So lipids are also composed of smaller subunits, although maybe not quite in the same way that the other three classes of biomolecules have monomers. So I think the best way to learn these is to kind of just jump right in. So, some of the main components of most lipids are fatty acid chains, fatty acid tails. So here is an example of one. Notice we have a big long chain of carbons with a whole bunch of hydrogens attached to the carbons. This we call a hydrocarbon chain. Makes sense, right? And then over here on the end we have a carboxyl group, which is one of those functional groups that you guys should know. One of the other major subunits of lipids is this glycerol molecule here. Notice it's composed of 3 carbons, several hydrogens, and then 3 hydroxyl groups here. So we can kind of combine these two things to make up triglycerides. So here is an example of a triglyceride. It is called that because tri, like a tricycle, meaning 3. There is 1, 2, 3 fatty acid chains, and triglyceride, because there is a glycerol molecule over here. See the 1, 2, 3 carbons, just like over here? So these two molecules, actually 3 of these plus 1 of these, are combined via, what do you know, dehydration synthesis. So here's kind of a close-up of that. The hydroxyl group from each carbon on the glycerol molecule and a hydrogen from each fatty acid chain forms one water at each of these locations. So 1, 2, 3 water molecules total will be removed to combine the fatty acid chains and the glycerol into a triglyceride. So triglycerides are some of the main energy storing molecules in the body. So if we need some energy, we can start to break down our triglycerides as well. So there are two different kinds of triglycerides. We have saturated and unsaturated. This one over here was an example of a saturated triglyceride. By saturated, we mean that the carbons in the fatty acid tails here are saturated or completely full with the maximum number of hydrogens. So the saturated triglycerides are what make up like your butters, things that are solid at room temperature. In terms of diet, the saturated triglycerides are sort of considered the more unhealthy of the two. These are the kind that contribute to things like cardiovascular disease. If we look over here, here's an example of an unsaturated triglyceride. It is unsaturated because of the double bonds between some of these carbons here. Remember that carbon likes to make 4 bonds. So if we are going to form a double bond, that means we had to break off some of the hydrogens, and therefore it is unsaturated with hydrogen. And these make up things like our oils, things that are liquid at room temperature, and in terms of diet, these are considered the more healthy of the fats. So let's see if we can find a triglyceride on our 3D molecules over here. Oh, and I see 3 big long chains, so that is kind of a giveaway that this is a triglyceride. And then, so our glycerol would be right over here. The next kind of lipid we're going to talk about is the phospholipid. So phospholipid is basically a modified triglyceride, but they contain a phosphate group, hence the name phospholipid. I like this image because it kind of shows you each of the groups here. So here is our glycerol, just like in our triglycerides. And in fact, here are 2 fatty acid tails, but then we have a phosphate group up here. So these are really similar molecules. It's just that one of the fatty acid tails has been replaced with a phosphate group. So phospholipids are kind of special because they are amphipathic molecules. Amphi means both. Think of like an amphibian. Amphibians like both water and land. So amphipathic molecules are hydrophobic on one end, meaning that they repel water on one end, and hydrophilic on another end, meaning that they attract water on one end. And this is kind of special because phospholipids make up a large majority of our cellular membrane. They actually form what is called the phospholipid bilayer, bi meaning 2. So here's kind of a zoom in on that. So this would be like the inside of the cell over here, and this would be like the outside of the cell over here. We know that our body is made up largely of water. So there's water, fluid, inside the cell and outside the cell. So the hydrophilic end, or the end that likes water, needs to be touching the water side. So this is like the phosphate groups here and down here. So that equates to that in this image. Now the phosphate groups, the hydrophilic phosphate groups, sort of arrange themselves so that the hydrophobic fatty acid tails are protected from the water in the middle of the bilayer. And this is sort of a image of what the cell membrane would look like, including some proteins and some other molecules in here. So let's find a phospholipid in our lab manual here. And there we go. Here's 1 fatty acid tail, 2 fatty acid tails. Here's the glycerol molecule, and here is our phosphate group. Now the next kind of lipid that we're going to talk about is cholesterol. So cholesterol is a molecule that contains 4 rings attached to a hydrocarbon chain on one end and a hydroxyl group on the other. So let's look for the 4 rings. This looks like it right here. 1, 2, 3, 4 rings. Here's the fatty acid, or I guess hydrocarbon chain on one end and the hydroxyl group on the other end. So cholesterol does a variety of things in the body. It helps maintain cellular membrane fluidity. It is also a precursor to all of our steroid hormones, and it aids in lipid digestion due to its presence in bile salts. So let's flip over to our 3D molecules and see if we can find cholesterol and phospholipid. So here is a phospholipid. We have 1 fatty acid tail and 2 fatty acid tails. Here is the glycerol, and here is our phosphate group. So our phosphorus, it looks like, is in purple. And let's look for our cholesterol, and keep in mind that these are 2-dimensional images of 3-dimensional models, so in lab you'll be able to kind of pick these up and move them around, which might be a little bit easier to tell what they are. So here is our cholesterol molecule. If you count it out, here's 1 ring, 2, 3, and 4, and here's a hydrocarbon chain and our hydroxyl group over here. Okay. And I think we hit all of our lipids. So let's move on to proteins. So proteins are the most diverse class of biomolecules, and they have many, many functions in the body. They help with transport, they help with immunity, they make up a lot of cell receptors, and a really big subcategory of proteins are enzymes. So get pretty used to seeing enzymes. So enzymes catalyze reactions, which means they speed up reactions. So they are very important in our body. We have enzymes everywhere. And what I mean by catalyzing reactions, that is lowering the amount of energy required for a reaction to take place. So enzymes, you can usually tell looking at a word that something is an enzyme because it ends in ASE. So remember, we saw OSE with our sugars, now we have ASE with enzymes. So proteins have pretty noticeable monomers or subunits, which we call amino acids. So here is the general structure of an amino acid. We have a central carbon here attached to a hydrogen up top, and then we have a nitrogen with 2 hydrogens on it, and this here makes up the amine group. And over on the other side we have carbon double bonded to oxygen, and then another oxygen and hydrogen, which makes up that carboxyl group, which is another one of those functional groups that you should know. Amino acids kind of get their names from the amine group and the carboxylic acid, amino acid. Then down here we have this R group. This R is just like a placeholder. It's a variable. And so between all of the amino acids, this R group will change, and that's what makes a specific amino acid that amino acid. So let's find an amino acid over here in our lab manual. And this looks like one right here. Central carbon, hydrogen up top, amine group over here, carboxylic acid or carboxyl group over here. And this would be our R group. Specifically, this one has a hydrogen here. And this is the most simple of our amino acids, glycine. Notice you have a list of amino acids over here, and the only difference between all of those is that R group. So I'll let you guys find the other two. So amino acids are joined together into big long chains via what's called peptide bonds. Peptide bonds are created via dehydration synthesis. So this image is kind of showing you, here goes a hydroxyl group from one amino acid, here's a totally different amino acid who's going to give up a hydrogen, H2O. Out pops a water molecule here, and now we have just joined together these 2 amino acids, at which point we call this a dipeptide. Di meaning 2, and the root pept basically means related to protein. So let's look for a dipeptide in your lab manual here. There we go. So keep in mind that wherever 2 lines meet in structural formulas in organic chemistry, carbon there. So here is our central carbon. Also, organic chemists don't always like to draw out their hydrogens, so it is assumed that you know that there is a hydrogen here. So central carbon attached to a hydrogen, just like we saw here, central carbon attached to a hydrogen. Here's our amine group, and there's another carbon here, the double bond to oxygen, which equates to this over here. And then this carbon bonds directly to the nitrogen in the neighboring amino acid, because remember this is a dipeptide, so 2 amino acids next to each other. So I know that we have a methyl group up here, so this is just an example of what this R group could be. So now let's flip over to our 3D molecules. And look for, first let's look for just an amino acid. She gives you some specific ones to know. We're just going to look right now for the general shape of amino acids. So remember, black is carbon, So